present a little bit. Okay, recording in progress. Got it, good, okay. So we just went through this agenda. So to discuss our emitter technology, so let me just give you a brief overview of it. Newomics M3 emitter for microflow LC and nanospray mass spec. So what that means is our emitter has the ability to take microflows in and they give out nanospray sized droplets at the exit of the emitter. A conventional emitter has a single stream that's shown here in the left-hand box, but the Newomics M3 emitter shown here has 8 nozzles spraying. So if you bring in 5 microliters a minute of LC flow into this emitter, it'll split it into 8 different streams at 625 nanoliters per minute. And the benefit of doing that, there's a couple of benefits. The most obvious is you're going to get smaller sized droplets, and it's going to enhance the ionization efficiency, simultaneously reducing the matrix effects, and it's going to increase your signal to noise. So that's a quick overview of the technology and how it works. The analogy is shown here of a shower head where you have a stream in and then a lot of fine droplets coming out. Now, if we look at Newomics ion source here, you can see, if we look at the source, it's a closed system. We have a camera built into it. So it's a microscope that looks and it's backlit to give you a nice image. You can see the image in the bottom here. This is what you'll see on your computer screen. It has emitter housing that's very easy to load and unload. No tools necessary for that. And then the fluidics connection is made here through a nanopiper. It's a very straightforward connection, and we provide a torque wrench so you get a precise tension to maintain the life of the fittings and not crush the emitter. So this system will deliver nanoflow sensitivity, which will give you lower limits of detections and limits of quantitations, allows you to use smaller sample amounts, and enables higher throughput at the microflow flow rates, and it achieves robustness for complex matrix analysis. And it's truly very plug and play, very easy to use. Now, if we look at the different mass spec interfaces we have thermo NG interface and we have thermo legacy interface. The big difference between these two is the way it attaches to thermo mass specs. We have up and down levers for the NG interface and side-to-side levers for the legacy interface. Other than that, they perform the same. And with these two interfaces, we cover the entire thermo product line. So any thermo mass spec can be used with our technology. And we developed this with Thermo. We have a co-marketing agreement with them. They helped us in the development of the product. And when you put the hardware onto your mass spec, the software will automatically recognize the source. And so there's full compatibility with all of our vendors. Also, we have the Bruker interface. And so that will work with all the QTOFs and timsTOF instruments. And that's been quite successful. And just introduced at ASMS of this year, we have our Agilent interface. So now we can interface with all of the Agilent instruments. Let's see. So here we have, I mentioned the nanopiper connection. And if we click on this, I can show you the video of the spray. So this is methanol flowing at 5 microliters per minute. Each nozzle here is 10 micron ID. We have a couple of different configurations of emitters. We also have a 20 micron ID nozzles. And this one's showing 100% methanol spraying. And you can see, you get a nice spray, nice huge sweet spot is generated. And then the mass spec pulls it in with both the electric field and the vacuum, pulls the ions right in. So that's works very nicely. Now, to cover the applications that we've developed with this. So we have application notes for all of these. And what that means is you have the proper voltage settings and other parameters to get you set up quickly with these applications. So we have proteomics, both top down and bottom up proteomics. We have intact, which is another reverse phase application. So we have intact mass. We have a number of people doing this with our flow splitting kits. So we have like a 10-to-1 flow splitter, and they can use regular LC to split. We have oligonucleotides. a few people doing nucleic acids. So we do both oligos and RNA as well. And then we have native mass spec is a very big application for us as well. This is with SEC, typically ammonium acetate buffers. We have a large number of people doing that as well. Native antibodies, antibody drug complexes, and even large molecules like AAV are being studied using our technology. And last but not least, we also have a lipidomics application for those of you interested in that. All right, so that's a quick overview of our technology. And now let me introduce our speaker for today. Jake Melby is a fourth-year analytical chemistry Ph.D. candidate in Yi's lab at the University of Wisconsin-Madison, where he's developing solutions to address the challenges associated with top-down proteomics. Jake's work has centered on connecting proteoforms with function, as well as developing high-sensitivity top-down proteomics methods for the analysis of proteoforms from minute biological systems. Currently, he is enjoying his stay out in the West Coast and in the Future Talents program at Merck in South San Francisco, working with the mass spectrometry group within Discovery Biologics. So today's program features Jake's presentation titled High Sensitivity Top-Down Proteomics Reveals Heterogeneous Proteoform Profiles Amongst Various Single Skeletal Muscle Fibers. So with that, I will turn it over to Jake and let him start the webinar.
All right, can everyone hear me okay?
Yes.
All right, and can everybody see my first title slide?
Yes.
All right, good call. Sounds good. I'm gonna get started. Hi everyone, my name is Jake Melby. I'm, as Tom mentioned, a fourth-year analytical chemistry student at the University of Wisconsin in Professor Inga's research lab. So we've been collaborating with Newomics for the last year or two, specifically working on this project that's titled High Sensitivity Top-Down Proteomics Reveals Heterogeneous Proteoform Profiles Amongst Various Single Skeletal Muscle Cells. So as Tom also mentioned, I'm actually out in the Bay Area this summer working at Merck. So I'm actually at Newomics site giving this webinar. So it's kind of like a dual virtual in-person type of a presentation for me. So let's get started. So before I get into the crux of my research, I wanted to give some context as to what our research program does in the Gut Lab at UW. So we take students from various different paths, such as biology, chemistry, and medicine. I'm a chemistry student, so I've spent a lot of my time learning top-down mass spectrometry, top-down proteomics, but we also have other students working on metabolomics, as well as biology students that are studying important things related to muscle, such as myofilaments, metabolism, GPCR signaling. And we also take medical students and collaborate with doctors on campus to study things such as heart failure and regenerative medicine, all under the lens of cardiac systems biology. But the one thing that connects us all within the group is that everyone is using mass spectrometry as the technology to study whatever the purpose of our project is. Oh, there we go. So while the Gut Lab's more famously known for studying cardiac muscle, I've spent a lot of my time studying skeletal muscle, which is a very abundant component of the human body. It makes up 30 to 40% of our body mass. It enables a wide range of movement, posture, and balance. So you can see this image of kind of a cross section of just a generic piece of skeletal muscle. The main point of skeletal muscle is to kind of accommodate all of these different demands on the body, but skeletal muscle is connected by tendons to bones, kind of to enable this movement. But it's made up of a lot of different components, such as tendons, nerves, blood vessels, fast and slow twitch muscle fibers, which are the individual contractile unit found within muscle, as well as several other things. And it's these proteins within sarcomeres within muscle that are performing all of the necessary contraction and relaxation and structural purposes in order to accommodate muscle. So mass spectrometry has proven to be a critical tool to measure the proteins found within these sarcomeres, which again, as I've already mentioned, is how muscle actually contracts. Here we go. So because this, people may have various backgrounds at this talk, there's two major ways to perform mass spectrometry-based proteomics. What's termed bottom-up proteomics is the conventional method where cells or tissue are taken and the proteins are extracted, digested using an enzyme such as trypsin into peptides, at which point liquid chromatography and tandem mass spectrometry is performed in order to sequence these peptides, at which point data analysis and database searching can match unique peptides back to a protein of interest. So the pros of using this approach is that it's well developed. You can get very deep proteome coverage, but some of the downsides to the bottom-up approach is that there's a lot of ambiguous information generated through this digestion process. So a lot of the molecular level information, such as post-translational modifications, isoforms, or if you're thinking in a drug world, drug binding, lots of important information is lost during the digestion process and following LCMS. The top-down approach is very similar, but it skips the digestion step. So again, we're starting with cells, tissue, whatever your biological sample of interest is, and extracting proteins of interest. But without digesting our proteins, we're keeping our molecules intact. So at this point, we can unambiguously characterize post-translational modifications and some of these specific things that make a protein a protein. So the pros of using top-down proteomics is that you're able to study combinatorial post-translational modification and sequence variants and quantify intact proteins. But the downside to using top-down is that it's underdeveloped compared to bottom-up. And due to this reason, the proteome coverage is often much less. But it still goes to say that this concept of a proteoform, or all of the different forms of a protein that can arise from a single gene, and this is due to events such as genetic mutations, alternative splicing, and post-translational modifications. All of these events can create what's been termed back in 2013 a proteoform. So kind of while top-down has its own challenges, it still provides you a lot of very detailed information at the proteoform level, which is critical in many different biological systems, but especially in muscle. It's been shown before that skeletal muscle contractile function is mediated by post-translational modifications of these proteins found within muscle, as marked by these stars. All of these proteins are modified by post-translational modifications, such as phosphorylation. And it's also been shown that isoform switching and altered PTM levels of sarcomeric proteins are implicated in many different things, such as muscular dystrophy, heart failure, aging, many different things. So while it may be underdeveloped, the ability to measure these biomolecules at the proteoform level provides a lot more useful information. So it's worth doing is what I'm trying to say. So the Gut Lab has spent much of its time over the since it began about 10 years ago, addressing some of these, what we call challenges to top-down proteomics. Mainly the big four that we've worked on is protein solubility, developed the top-down mass spec compatible surfactant, proteome complexity. We've developed various multidimensional chromatography methods for intact proteins, dynamic range, which we've addressed using nanomaterials to enrich low abundant proteins, as well as the software side of things. We've developed an in-house software that's free and user-friendly. But what I've spent a lot of, and many of these challenges are summarized in these two reviews if people want to dive more into the details of some of these challenges. But what I've spent a lot of my thesis working on is sensitivity. So sensitivity is definitely, it's definitely something that's very important for top-down that isn't necessarily talked about all the time. So I'm going to get more into this in a few slides, but that's essentially what the context of my thesis is, as well as this talk. So previously, we've shown that top-down proteomics can be used as a robust and high-throughput method to characterize the molecular heterogeneity of myofilament proteoforms from 7 different rat skeletal muscle tissues. So this was a project that I was involved in my first year in graduate school, where we were taking 7 different pieces of skeletal muscle tissue from a rat hind leg, performing a 2-step protein extraction to get the sarcomere proteins of interest, and then performing online LC-MS to characterize the different proteoforms found within these muscles. And what we found was that there's distinct proteoform profiles observed from each tissue extract, as shown by top-down LC-MS. And you can see this both chromatographically as well as in the mass spec data. Chromatographically saying that all these peaks look a little bit different, some of them similar across the 7 different skeletal muscle tissues. But what's really quite interesting is an example, troponin T3, which is an important member of the thin filament of the cardiac or skeletal muscle sarcomere. It has a pretty remarkable amount of proteoform heterogeneity found from tissue to tissue. But one of the challenges with performing proteomics measurements at the tissue level is that your results are an ensemble measurement of all the different components found within muscle. And then a few slides ago, I showed that in muscle, there's a hodgepodge of different things, such as blood vessels, nerves, fast and slow twitch, single fibers. So one of the difficulties here is that these measurements that we see in this slide is they're coming from a 5 to 10 milligram piece of tissue, which contains a very heterogeneous population of components inside. So our measurements, and this is true for any tissue measurement, is again more of an ensemble measurement of everything that's going on in the system. it would be extremely beneficial to be able to do this at the single fiber level, which is where I'm headed. So single muscle fibers are the contractile apparatus within skeletal muscle tissue. They're elongated and multinucleated single muscle cells that serve as the way that muscle contracts. So they're really small, 1 to 2 millimeters in length and 200 microns in width. This is actually a microscopic image that we got in the lab of basically when you get skeletal muscle tissue, then you get basically to this level of the tissue, and then you're able to start pulling these fibers apart under a microscope and then placing them in a tube. And using a nice enough microscope, we can actually start to see some of this multinucleation, as well as some of the muscle striations found within the muscle. They're classified as fast and slow twitch fibers, which determines which isoforms are expressed as well as their function. And PTMs and isoforms of these proteins have really, really important effects on the function of the fiber. So the challenge with doing this through a top-down lens is that they're really small. Basically what we need to think about is a way to extract the limited amount of sample from these single muscle fibers and get as much as the sample to a mass spectrometer as possible. So there's a few things that you really need to think about that are always true in proteomics experiments, but accentuated when doing this at the intact protein level. So the first, and I would argue the most important, is sample preparation. So sample losses occur. This is normal whenever you're extracting proteins from a sample of interest and then subsequently working up that sample to be mass spec compatible. But this is particularly an issue when you're working with sample limited systems such as single fibers or single cells. Essentially, the amount of starting material you're starting with is so much lower that any amount that you lose has a much greater effect than if you were starting with a lot of something and losing that same amount. I was kind of thinking about it on the way up to Berkeley today. It's almost like if you were at a casino and you had like a lot of chips and then you threw in one chip, you know, you still have 99% of your chips. But if you're me and a bad gambler and you only had 2 chips on the other end of the table and you threw in 1 chip, well, now you have 50% of your chips gone. So that's essentially what this figure to the right is also showing. The black bar is showing an 1,000 cell equivalent starting from 1,000,000 cells and the number of peptide IDs. And then starting from 1,000 cells alone, you can see the number of peptides is almost none. So all that to say is that sample preparation is critical. Finding ways to minimize sample preparation steps is going to go a long way. Due to the complexity of the proteome, you need to do some sort of front-end separation before you do your mass spectrometry measurement. Again, this is normal, but this is just another opportunity for sample to be lost, such as on the column. And it's another important consideration that since you're starting with such little sample, it's going to be difficult to do multidimensional chromatography. Lastly, proteoform characterization. Excuse me. So the coelution of low molecular weight proteins, excuse me, it's necessary to fractionate small proteins from large proteins due to the exponential signal to noise decrease as mass gets larger. So all that to say, for sample limited systems, proteoforms are detected, are going to be mostly high abundance and low molecular so that brings me to the design of the method. So we're starting with the rat, using the hind limb as well as 3 different muscles. We're pulling single muscle fibers under a microscope and using 2 different populations, one for force measurements, so using a force apparatus to measure the force at which these fibers contract, as well as using a group of these fibers from each muscle to perform online top-down proteomics. So first, I'd like to start with the force measurement data. So this was done in collaboration with Gary Diffie. He's in the Department of Physiology on campus. And essentially what he did was he was able to get single fibers into this force apparatus instrument and able to apply a frequency, which essentially makes these fibers contract, and their shortening velocity is correlated to the function of the fiber itself. So as you can see from the data, from each of the muscles, there are different shortening velocity characteristics. So this is more of a big picture measurement, force, kind of functional measurement to kind of help guide our proteomics data. So now that we know that we have 3 unique sets of fibers from 3 different muscles, we were able to perform top-down proteomics on a separate population of the fibers. So we pulled the fibers underneath the microscope, placed the fiber into a low-bind tube, and then using mass spectrometry compatible solvents performed a freeze-thaw lysis due to the fact that these fibers are so small that you can't mechanically homogenize them in any way. So essentially what we're doing is we're getting the fiber into a mass spectrometry compatible solvent and then performing a freeze-thaw lysis in order to solubilize all of the contractile proteins within the single fiber. At this point, we're loading 100 nanograms onto a thermal MabPack column, C4 column, that we're operating at 2 microliters a minute. And this is connected to a Newomics MnESI source with its 8 nozzle, 10 micron emitter. So this, as I'll mention in the next slide, helps us with, it's kind of a sweet spot for top-down separations with the sensitivity of nanoelectrospray. All this work was done on a Bruker Mass 2 QTOF, and the data analysis was done in Mash Explorer. So first, I wanted to talk about the Newomics Microflow Nanoelectrospray Ionization Emitter, or MnESI. So as Tom showed earlier, this is kind of an image looking at the camera from the lab itself. You can see the nice Taylor cone, nice electrospray plume from the 8 10 micron emitters. So when I first got the source and emitters and was using it myself, I was just performing in LCMS using a protein standard, which was carbonic anhydrase. It's about 30 kilodaltons, so it's a great standard to work with for top-down proteomics. And I was just doing a baseline characterization kind of comparison with our previous ESI source. And as you can see from the extracted ion chromatogram, we're getting about a 5-time signal increase in our signal. And it's also eluting first and sharper. So the signal increase is due to the nanoelectrospray effect. The fact that it's eluting first and sharper is due to the fact that the dead volume in the Newomics source is about 20 nanoliter, whereas in the other ESI source, it was about 1 microliter. So our peaks are eluting a little bit before and sharper, which is also a benefit when you're doing top-down proteomics in separating a lot of peaks from one another. So again, the signal is seen at the MS1 and MS2 level as well. And when you actually get down to looking at the fragment ions, there's about a 63% increase in sequence coverage when we use the MnESI source. And a lot of these fragments are some of the more difficult to resolve ones in the middle of the spectrum, or middle of the sequence, that the signal to noise is usually too low to accurately assign these fragments. But through our boost in signal, we're able to confidently assign those fragments. So yeah, now this is like getting to the actual data from the method I described. These are 3 different base peak chromatograms from the 3 different muscle tissues. I have it kind of labeled here because there's a lot of acronyms and jargon going on around here, but essentially all of the labeled proteins in this chromatographic trace can be seen are most of these very important contractile and structural proteins found within sarcomere. And you'll see soleus, if you remember from the functional data, had the slowest shortening velocity. So soleus is most correspondent to the highest population of slow twitch fibers. And you see that in the data is proteomics data as well, is that many of these contractile proteins are in their slow isoforms, which you can see as slow skeletal troponin I, beta tropomyosin, myosin light chain 2 in its slow isoform. Whereas the VL, or the quadricep muscle, which had the fastest shortening velocity, you see a lot of these proteins in their fast form. So again, it's an interesting way of kind of looking at things at a functional big picture level, as well as at the protein level. So now I want to kind of go through and look at some of these at the mass spectrometry data in detail from fiber to fiber. So skeletal troponin T, just to give some context to what these proteins actually do, it regulates the calcium-mediated interaction between myosin thick filaments and actin thin filaments during muscle contraction and relaxation. And this is a protein that has a diversity of isoforms expressed in skeletal muscle. And each of those are for different effects on sarcomere contractility. So what we're looking at here, red is the VL, so the quad muscle. This is the fastest functional data. Plantaris is this muscle in the back of the calf. This had more mixed characteristics from the functional data, as well as the soleus in blue, which is the slowest. So essentially what we're looking at here is the phosphorylated forms of these proteins, as well as the unmodified and loss of water. So P total is a calculation that's essentially quantifying the amount of the phosphorylated form of a proteoform family. And at this level for fast skeletal troponin T3, the phosphorylation is relatively consistent. And it's about 0.7. Whereas for the soleus fibers expressing slow skeletal troponin T1, the amount of phosphorylation was a lot higher. And you also see a larger diversity of proteoforms here. So that was for fast skeletal troponin T3. Not a ton of diversity in the PTMs observed. And this was for the most abundant form of the troponin Ts. But when I was looking at the deconvoluted spectra, and you kind of zoom in on this region that's in the box, we start to see some low level troponin T isoforms as well. So I'll kind of navigate everyone through this figure, but we've zoomed in 20 times from this spectrum essentially. And now we see the presence of fast skeletal troponin T4, as well as its phosphorylated form, fast skeletal troponin T9, troponin T10 in its phosphorylated form. And what's really interesting is there's a remarkable amount of heterogeneity in these lower abundance proteins. So to the point where some of the proteoforms exist in some fibers, but not others, such as what I'm putting my cursor over here and here. Whereas in the quad muscle, it was relatively, they all had the same troponin T4 proteoform as well as the phosphorylated variant. But a lot of the diversity came about in the plantaris muscle. All of this is quantified in the bar chart here. So again, the higher abundance proteins of interest, relatively consistent, lower abundance, showing a pretty remarkable amount of heterogeneity, both from fiber to fiber as well as tissue to tissue, showing differences. The next example to kind of illustrate this proteoform heterogeneity is myosin light chain 2. So myosin light chain 2 is known as the regulatory light chain because it's actually the phosphorylation of myosin light chain 2 residues that promotes myosin head movement towards actin filaments. So it's basically controlling the molecular motor of muscle. So obviously measuring this PTM is extremely important when understanding muscle biology. So again, when we look through this data without even having to kind of understand what's going on with the data, we can see that there's a lot of differences in our plotted data. So I'll take, for example, the P total here of 0.05. So this indicates that for this fiber, there is a very low amount of phosphorylation, whereas a fiber, again, pulled from the same piece of muscle, same kind of bundle of fibers, can show as much as a 6-fold difference in phosphorylation. So that's what's quantified here and here. Whereas for the quad muscle, you can just tell visually that the values are more consistent, relatively quantified around 0.15. And then for the plantaris muscle, again, showing some pretty remarkable heterogeneity from, for example, 0.22 to about twice that of 0.4. So again, to kind of pitch why it's important to be doing this at the single fiber level, even if I had obtained a sample that was just 6 single fibers, I would essentially be taking an ensemble measurement of all 6 of these components and getting a measurement somewhere in the middle. Whereas it could be very important to be correlating these PTMs of interest at the fiber level instead of at the tissue level. So thus far, I've mostly just showed MS1 level data, but top-down implies that we're doing tandem mass spectrometry as well. So using these same myosin light chain or regulatory light chain isoforms, you can see by the green that there's a lot of sequence homology. So with bottom-up proteomics, you need several confidently identified peptide, tryptic peptides or other types of peptides in order to get confident protein identifications. I haven't gone through the computations, but obviously this is a very highly homologous 2 proteins. So there's a lot of benefits to be able to disseminate the two of them without the need of tryptic digestion. Because as you can see here, we're able to separate these proteins with liquid chromatography, so we can effectively quantify the area under the curves if we want to do isoform quantitation. At the mass spec level, we're able to select the precursor of interest with high mass accuracy and perform collisional-based dissociation to get B and Y ions, and unambiguously characterize these very highly homologous and important proteins of interest. So it speaks to the benefit of being able to do this at the intact protein level compared to doing this at the peptide level. Let's see. So the next protein of interest I wanted to talk about is myosin light chain 1. It's known as the essential light chain because it's important for the structural stability of the myosin head. And in skeletal muscle, there are 3 different isoforms found. So in the VL and TLN muscle, the fast isoform was expressed, whereas in the soleus fibers, it was found that there was 2 different forms of the slow isoform being expressed. And again, we're doing this at the intact protein level, and our mass spectrometer is high enough resolution that we're still getting nice baseline resolution for these 20 to 25 kilodalton proteins. Again, now we're dealing with 3 highly homologous but important proteins of interest. We're able to separate all 3 of these. You can kind of see how sharp these peaks are. They're on the order of less than 30-second peak width. That's largely due in part to the phenomenon I described earlier with when I was first comparing the data to the ESI source, is that these elution peaks are really small, and that's definitely in part due to the low dead volume of the Newomics MnESI source. So yes, again, these are all separated chromatographically, able to select all of the precursors of interest and fragment them to, again, unambiguously characterize these proteins of interest with very minor differences in their N-terminal modification, and more or less like almost homologous from the middle of the protein to the C-terminus. So all that to say is that it top-down unambiguously characterizes highly homologous MLC isoforms, and this is all from a single MSMS scan. So I could go back and average some of these scans to get even higher sequence coverage, but I think it's powerful just from a single scan to say that we have all this information. The last example that I'm going to go and talk about for this one is myosin heavy chain. Myosin heavy chain is a massive protein. It's 223 kilodaltons. This is where the other 2 proteins 2 of the other proteins I've talked about modulate the motor and neck of this molecule. It interacts with the thin filament actin to convert chemical energy to mechanical energy, and that's what powers muscle. And this is actually how single muscle fibers are classified historically, is based on the isoform expression of myosin heavy chain. So for slow twitch muscle fibers, they're called type 1 due to the isoform of myosin heavy chain they express. And then for fast twitch muscle fibers, there's 3 different type 2 isoforms that are expressed. So usually we do not see this protein in our conventional workflows, usually because it coelutes with actin, which is about a 40 kilodalton protein. So much easier for top-down to manage. But again, due to the fact that my chromatographic peaks were so sharp, I actually was able to get several scans consecutively of just myosin heavy chain as it was coming from, well, off of the LC column through the Newomics source. And upon doing a low resolution deconvolution, I actually found that there were fiber-specific isoforms being expressed in these single muscle fibers obtained from different tissues. So going back to literature, we'll start with soleus, which is 97% type 1, so 97% slow twitch muscle fibers. And more or less, we're measuring one mass for the soleus muscle fibers. So this is most likely the myosin heavy chain isoform associated with type 1, whereas for VL, the fastest of the bunch, it's 96% type 2B, which looks like we're more or less getting one measurement from these fibers. And then the plantaris, which is our mixed fibers from the mixed tissue, has a variety from type 1 to type 2 fibers. And again, we're kind of seeing that here in our low resolution data, is that there are some at different masses, essentially saying that, you know, there's most likely different myosin heavy chain isoforms being expressed from fiber to fiber within the plantaris tissue. So usually for top down, proteins above 50 kilodaltons become very difficult to analyze. And this is all just MS1 at this point. But I only loaded 100 nanograms of sample, which is pretty low. We were just using a quadruple time of flight as a mass spectrometer. So I think in the future, with the use of some smaller scale SEC, and we have a 12 Tesla FTICR in the lab that we very well may be able to get some of these better characterization data for myosin heavy chain, which is a critical protein in heart and skeletal muscle. Yeah. So in conclusion, we developed a high sensitivity top-down proteomics method for the analysis of proteoforms from single muscle fibers. Traditionally, biochemical assays will dedicate single fibers for single measurements. For example, you may have a group of fibers to run an STS-PH gel. You may use a group of fibers to do a Western blot on a PTM of interest. But with this method, you're getting all of that information from the same run. The data show that there's distinct differences in PTMs and isoform expression across the different muscles, and that there are fiber-specific isoforms of myosin heavy chain that we detected for the first time from single muscle fibers from the intact lens. So in the future, we're hoping to use this method to connect proteoforms with function to better understand muscle-related diseases at the single fiber level. So being able to perform functional measurements and proteomics measurements using the same fiber to kind of correlate changes in PTMs to the way fibers behave is kind of where we're headed with this. So with my remaining time, I just wanted to briefly talk about other projects we've been working on in the lab using the Newomics source. So it's a large goal of our lab, as well as it seems like Newomics is the precision medicine idea, which looks at the genetics, environment, and lifestyle of a person in order to select the best treatment for them. So in our eyes, where we're studying heart disease largely, we would love to be able to get to the place where we can take a patient's sample, tissue, blood, other body fluid, and study this sample directly, as well as create patient-derived cardiac cells so that we can study a patient's heart characteristics in a dish. And we really think that proteoform measurements are going to be a critical part of enabling precision medicine because it's really the proteoform that's the functional actor of the cell. So one of the projects we've undertaken previously in regards to precision medicine is studying hypertrophic cardiomyopathy, which is the disease of the sarcomere, as 1,400 mutations have been identified in at least 11 sarcomere genes. And basically what we showed very quickly is that regardless of the HCM mutation, there is a convergence at the proteoform level. But one of the difficulties with this project was that these septal myectomy tissues, which is a surgery performed to correct the obstruction that's impeding blood flow, and this is the sample that we studied it in this work, is very small, and it was limited to one-dimensional LCMS methods. So we're hoping to use some of these concepts here, such as serial size exclusion chromatography, more narrow SEC columns to limit sample consumption, as well as the high sensitivity denatured Newomics method I described previously, to be able to do multidimensional chromatography offline from these small pieces of patient biopsy samples. So that's denatured mode, but we're also hoping to perform this in native mode, where we're taking our SEC columns. This is working with Andy Alpert at PolyLC, where we're taking our SEC columns using a Newomics flow splitter and then directly analyzing these native complexes from these tissues, because a lot of the proteins I've been talking about in this talk are actually found in the complex. So that's something, all things that are in the works. And lastly, I briefly mentioned our nanoproteomics project, but we've published a paper in Nature Communications where we're analyzing troponin I from serum, getting down to some of these detection limits. But now it's our goal moving forward to make the limit of detection even lower for cardiac troponin I, and eventually use this nanoproteomics technique, which is providing proteoform level information, to replace traditional biochemical assays such as ELISAs, which is more a measurement of just light and concentration. Hoping to do this in the future. I've already started some of the preliminary work, and again, I think using the Newomics source is going to prove to be a really critical part of this method. So yeah, that's what I've been working on and what we plan to work on. With that, I'd like to thank Ing, who's my boss, great mentor and kind of helped with all this work. Big thanks to Newomics for collaborating with us and helping out along the way, as well as Bruker. And thank you all for your attention. I'll be happy to take any questions.
Okay, great. Thank you, Jake. That was great. We really appreciate that. So let me read some of the questions we got here. Now that you've done single fiber, what do you think you're going to need to get to for a detection limit to do single cell proteomics on these molecules?
Yeah, no, that's a very good one. So I think it comes down to sample preparation again. So single fibers are single cells, but they're large single cells. So I'm able to use kind of sample prep. I more or less use a one vessel sample preparation and just your normal low-bind tubes, try and do whatever I can using developed cheap technology to minimize sample losses. If I was to move to single cells, I think I would need to get a little more sophisticated in the sample preparation side of things. I would need a cell sorting device. I'd need to find a way to sterilize the cell. using top-down mass spec compatible solvent. And then I'm currently using a 150 micron ID column. I think a more narrow column is going to help a lot, such as like the NanoMia, which is some of the newer technology at Newomics. But I think if we could get that in a microflow version, that would be a huge step towards doing top-down on a single cell. Because I mean, I think it is a lot of people's goals to be able to get to that point. Even PNNL has published a paper using the nanopods to do this. So I think a lot of people are thinking about it. And the way I see it is that over half of it comes down to sample preparation.
Okay, great. Next question is, how much did the emitters cost? I can answer that one. They are $349. And how long do the emitters last, Jake, in your hands?
Yeah, so I can only speak to my experience. And I guess there's a lot of downsides to working on sensitivity in that there's not a lot of your sample, but one of the benefits to working on sensitivity is I never clog a column. I've rarely clogged, there's nothing really there, so there's nothing I really ever contaminate. So that was a long-winded way of saying my sample's pretty clean. I've used emitters for, I'd have to go back and look at my sample log, but I've used them for several experiments before with no problem. Yeah. So I think for my application, denatured, top-down, limited sample, I've been able to use an emitter for weeks at a time. I can't speak to native. I've never done native with it, and I can't speak to like serum or some of the more like high concentration protein work.
Okay, great. All right, thank you, Jake. All right, last chance for questions. Enter it in if you've got any. And I think that's it. So it looks like we'll wrap up right around 2 o'clock on schedule here. Thank you, everyone, for your attendance. Really appreciate it.
Thanks, everybody.
Thank you.
All right. Well, that was great, Jake. Thanks.
Yeah.